All right, good day, and it's really wonderful that we have this opportunity to get together in our isolation. So I want to first talk about what a protein is, and this is something that my colleague Nathan has insisted that I start every NASA talk with, which is proteins are polymers of amino acids. So by polymer, I mean that this is essentially a chain. It's a chain of groups, the amino acids, and these amino acids are what determine the structure and the function of the protein. And these chains, like you see here in the upper corner of the screen, these chains will adopt a three-dimensional structure. The way that evolution acts on proteins is to change the amino acid sequence, to change the order of these monomers in the polymer, and a small percentage of sequences that are possible will fold into highly organized three-dimensional structures such as this. So you could imagine if this protein here is hemoglobin, each of the domains in hemoglobin is made up of a chain of about 150 amino acids. So if that chain, let's say for imagination's sake, was about 10 feet long, then hemoglobin itself would be about the size of a large cantaloupe. So these are actually very compact structures that these chains are forming. And the ability of amino acid sequences to form these compact structures makes amino acids part of a privileged set of polymers that can fold into complex forms. These polymers that can do this are called foldimers. Proteins are one, RNA is another, and there are a few, not many, a few synthetic polymers that can do this as well. So this compact structure that you're looking at over here that's moving back and forth, this is the molecule hemoglobin, which transports oxygen in your body. It's also involved in some redox reactions, including nitric oxide regulation. If you were able to measure this protein, it would be about 60 angstroms, 6 nanometers across. So it is really a machine that functions on the nanoscale. In addition to binding and releasing oxygen, it can sense the pH of its environment. It can bind carbon dioxide, nitric oxide, do a number of other chemical reactions. So it really is a complex multifunctioning machine. And over time, the sequences of proteins have evolved to adopt these three-dimensional structures because it is these structures which give proteins their function. Another thing which has really allowed proteins to enhance their functional potential is the ability to complex small molecules that are available in the environment or that are synthesized by the cell. So for example, in this case, at the center of one subunit of hemoglobin, we see this red disk over here. That's a porphyrin, a chemical group that's being shown over here. And at the center of that is an iron atom. And that iron atom is the active site, the functional site of this particular protein. And all of the other 5,000 or 6,000 atoms that make up this protein are really functioning to control the reactivity of this one site. So this is a protein that we have today. And as Paul had mentioned, really the goal of theme one is to understand how something as elegant and complex as this could have evolved from simpler forms. All right, so this is a sort of a systematics attempt to classify the folds of proteins. So remember, the fold of a protein is the way in which the linear chain adopts a three-dimensional structure. And it's that three-dimensional structure, along with metals and other molecules that the protein binds, that gives it its function. And this is just a small subset of the folds that are available in the protein data bank, in the PDB that Paul mentioned. The PDB is really this vast menagerie of proteins with all kinds of different structures. And what's amazing is that with the same sort of alphabet of the 20 amino acids, we can make all these different morphologies, all these different folds of proteins. So in this case, what we're seeing is a number of different proteins that are they're being separated based on some parameters of their structure. So you may notice if you look closely that some of these have these tight coils, some of these have these arrows. These are what we call alpha helices and beta sheets. And here they're separating these proteins based on the number of sheets, the number of helices, et cetera, that they have. So it's an attempt at a systematics of proteins. But this doesn't necessarily give us evolutionary information. There's a lot of systematic classifications of proteins that structural biologists such as myself use, but to really understand the evolution of these proteins, we also need some sort of a model to explain how they evolve. And so this model, as I'm sure most of you are familiar, is inference of evolution from inheritance, that there's this passage of traits from one generation to the next, and with each generation, there's the possibility of mutation or recombination or other changes to the sequence of the protein that could then change its function. But there's always this lineage of proteins that traces back to some common ancestor. And there are a number of trees such as this, some that are for the sequences of proteins, and it's used to understand their evolution, also for the morphology of organisms, and you can use this to produce species trees. For proteins, it's actually very challenging to make to make inferences based on structural similarities between proteins, and this is a problem that that our group, along with Paul and Yana and Nathan, have been working on now for over a decade, which is how can we build an evolutionary tree using the structures of proteins. Sequences can change relatively quickly. We expect that structures are more strongly conserved, but building trees based on structure alone, there are a number of methodological problems with that I can go into if anybody has any questions on that. So this is really the problem that we wanted to attack, which was, can we come up with a model for inferring what the smallest units of proteins were that were at the root of this evolutionary tree? And of course, we know that the proteins that we have now are these complex nanomachines, and they're very sophisticated in their structure, and that these are not the proteins that were there at the very start of life. There had to be much simpler, smaller forms of proteins that gave rise to the proteins that we have today. And our models of inference that we currently have are limited in our ability to look back in time. I'm going to describe to you the approach that we took and then show you some of the insights that we've gotten into protein evolution from using this approach. So here we're looking at an enzyme called hydrogenase. Hydrogenase is an oxidoreductase, as Paul described. It couples electron transfer with some catalytic activity. And in this case, it takes 2 electrons and 2 protons, and it makes hydrogen gas. So it's effectively reducing protons and making molecular hydrogen. And there's a large protein matrix composed of amino acids, which are the light blue and the light green here. But then in the center of this protein, there are a number of these small molecules, kind of like the porphyrin that hemoglobin bound. Here, this protein hydrogenase binds iron sulfur clusters, which are these yellow and orange structures here, and then a nickel site over here, which is the actual catalytic site. And we know that the electron transfer itself was likely a critical aspect of the function of this protein. So the approach that we're taking is, although we have information about the entire protein and the structure of the entire protein, we're really only going to be focusing on the structure that's in the neighborhood of the metals and organic cofactors that can transfer electrons, because these are likely the oldest part of the protein. These are the ones that were originally carrying out the function, and these are the ones that are going to be most relevant to the evolution of metabolism, because they're the ones that are carrying out the bioenergetic processes. So after playing with several different ways of doing this, we came up with a model where we would consider, as Paul used the word Lego blocks, we would consider each metal site to be a Lego block. And we took all of the proteins that were available in the PDB, in the protein data bank, that contained metals. This was about 10,000 proteins for which we had structural information. Many of them had multiple metals. So in the end, we had about 30 to 40,000 metal sites. And we then took these and classified them based on structural similarity. And so that looks something like this. We took a bunch of different proteins, we broke them up into smaller pieces, and then we took pairs of pieces and aligned them on each other, tried to see what were some components of proteins that would show up again and again and again. And what we found was that there were thousands of different kinds of metal sites in proteins, but there were a few that showed up again and again and again in lots of different proteins that would show up in a hydrogenase that could reduce hydrogen, in a nitrogenase that could reduce nitrogen. And these parts of proteins were found in lots of different metabolic pathways. For example, here we have this helical structure that binds two irons. Here's a mostly beta sheet structure that binds copper. Here's one that binds another porphyrin, and then an iron sulfur binding mode. And what we inferred from this is that these smaller pieces of structure, because we see them again and again and again, are likely ancient. These are the building blocks that were co-opted by lots of different enzymes. And really the way that nature has made complex enzymes from these simple ones is by just connecting Legos together and building an electron transfer pathway that acts as a wire, or acts as a diode, or acts as a transistor, or so forth. And it's these electronic circuits within these pathways that controls the flow of energy. So we had now all of these pieces. The pieces themselves, this is still sort of a systematics approach. We're not saying anything yet about the evolution of these pieces. All we're saying is that these are the different types of pieces that we have. So the next thing that we did with this data, and this is actually coming from more of an engineering point of view rather than an evolutionary point of view. We were previously funded to actually build electron transfer proteins out of these pieces. And so in addition to identifying what the individual pieces were, we also wanted to understand how are they connected together? How are they assembled into circuits inside of these proteins? And what we found was that if you look, for example, at hydrogenase, what you have is really, you can see it visually, and we can also quantify this. You have a very clear wire going through the center of the protein, where the individual metals, which are the transition points for electrons to move through, are separated by a small distance. The longer that distance is between metals, the less efficient electron transfer is. That's what this equation down here describes. And so there was a critical distance by which metals had to be separated in order for there to be effective electron transfer. So we went back into that data set of proteins, and now we asked for all these different folds of metal coordinating domains that we have found, how are they connected together? And can we learn something about how these connections are used to build more complex proteins? And this data set over here, this is really, I think, the meat of this particular analysis. This is where we've learned the most about protein evolution from looking at this network. And I'm going to talk about it in a couple of different ways. So let me give you a description of what you're looking at over here. So each of these nodes represents one classification of a metal binding fold. So this yellow one right here that I'm mousing over right now, this is a ferredoxin fold, a bacterial ferredoxin fold. It typically binds a 4-iron, 4-sulfur cluster, and it has a topology composed of 4 of these beta strands, those arrows, and then 2 alpha helices. The yellow color here means that it's binding iron and sulfur in some form. Over here, for example, this is a cytochrome C type fold. It's binding a porphyrin. This is a Rossmann fold. It's binding nucleotides and then often some electron transfer competent organic cofactor, like a flavin. Up here we have some folds that bind copper. So each spot that you see on this network represents a different fold based on our method of structural classification. And then the lines here represent pathways of electron transfer. So for example, we see that ferredoxin, this ferredoxin module right here, has a connection to fold type 128, which means that in some protein, somewhere in biology, there's a structure where a ferredoxin fold is within a cutoff distance of fold 128. And we imagine that electron transfer could happen along that pathway. And so we aggregated all of these electron transfer connections into this network. And remember, the original idea here was to use this to devise rules for assembling synthetic electron transfer proteins. But as soon as we saw this network, we started to see interesting patterns. All of these connections in this network are for function. These are electron transfer pathways. This is not an evolutionary tree per se. These are not connections that are explicitly defined by inheritance. But what we noticed was very interesting was that, for example, this ferredoxin, which is yellow, is connected to a lot of other types of folds which also bind iron sulfur, meaning that you often see ferredoxin, which itself has iron sulfur, connected to another fold, which, while it doesn't look like ferredoxin in terms of its structure, has a lot of the same chemistry. So this is a neighborhood of similar chemistry right here, right? Same thing with these porphyrin binding molecules up here, these hemes. We see this neighborhood of hemes. We see a neighborhood of copper. You see a neighborhood of organic cofactors. And, if I were to have an outlet in my house and I were to see, I could plug in a toaster, or I could plug in a computer or a television or my hair dryer. That's kind of what these connections are saying. I can take ferredoxin and I can plug all of these different things into it, and it'll transfer electrons to them. But what's interesting is a lot of the things that I'm plugging into ferredoxin look a lot like it in terms of their chemistry. And so one of the ways that we are interpreting this is that these connections, not only do they represent a functional connection, but a functional connection in the metabolism of electron transfer, but they also represent an evolutionary connection. That over time, this ferredoxin domain may have duplicated, and a second ferredoxin may have diversified its structure and changed to the point where we can no longer recognize it as a ferredoxin. And so this is what we consider to be extreme sort of duplication and diversification. And you can see, for example, a number of these nodes have these self-connections, and that is sort of a more traditional domain duplication. So for those of you who are not biologists, one of the ways that proteins grow from being small to being bigger is that a domain of a gene will copy itself, and then you'll have tandem 2 copies of that gene. And when that protein is expressed, now instead of having one cantaloupe, you now have 2 cantaloupes that are connected to each other. And that's now the larger, more complex protein. And what's happening here, we believe, is that second cantaloupe, over billions of years of evolution, is changing into an orange or changing into some other fruit. We still recognize it as a fruit, but it doesn't look similar enough to the original to recognize it as a cantaloupe. So pardon the analogy, but that's how we're interpreting this. So we think that in this functional network, there's the potential to make evolutionary inference. The second thing that's very interesting is at the core of this network are these 2 folds, the Rossmann fold that I mentioned and the ferredoxin fold. And both of these are very reducing, meaning that they have a very low affinity for electrons. They want to donate electrons to other molecules. And what we know about the evolution of the geosphere over Earth's history is that early in Earth's history, electrons were abundant. And molecules such as flavodoxin, for example, or ferredoxin, they would be able to take electrons from the environment because there were a lot of high energy electrons that were available. There weren't a lot of places for those electrons to go. And so metabolism early on was very limited by the electrochemical properties of these early proteins. But then we know over time, the geosphere has become more and more oxidizing. And as we look from the center of this network out, we see that the electrochemical properties of these units, of these folds, also become more oxidizing, meaning they're more likely to accept electrons. They have a higher affinity for electrons. So we see this gradient in the network from the center to the outside, which parallels the redox evolution of the Earth. And so this suggests now a time axis from the center of this network to the outside, where the earliest domains are in the middle and the newer domains are on the outside. And then one of the things that we noticed was when we looked at this network, there were a number of connections between these 2 early domains. Now, this could just be plugging a toaster into an outlet, and there's no direct evolutionary connection between the outlet itself and the device that I'm plugging into it. But what we noticed was that when we looked at the ferredoxin fold, which binds iron sulfur, and then these Rossmann folds, which will either bind a nucleotide cofactor or an iron-sulfur cofactor, they both contain the same repeat of elements: a strand, which is shown here as a arrow, a helix, and then a strand. So in ferredoxin, that's repeated, and you get this doublet of that motif. In the Rossmann folds, what you see is that it's repeated, but then there's an insertion of another helix in here. So this led us to hypothesize in our most recent study. This is still speculation that we're testing in a number of different ways, but that both of these folds actually had a common ancestor, which is a small peptide that looks something like this, where it's just a beta strand, an alpha helix, and a beta strand. And that could either bind nucleotide cofactors or metallocofactors. And that led to the evolution of these two large classes of protein folds that we have today. So we're using this functional network of electron transfer to make evolutionary inference, and then we're testing these ideas in the laboratory. So now I'm going to move over to some of the ways that we're testing some of these models of evolution using molecules in the laboratory. All right, so we've been very interested in ferredoxin for a long time, and our group and a number of others have flagged this as likely a very ancient protein fold. In proteins, even those that don't have metals, this particular topology of alpha helices and beta sheets is commonly found. Modern ferredoxins, even the ones that we have today, you can clearly see that there's this symmetry in their structure, that there's some that there was probably a duplication of a strand helix strand that led to the formation of the larger complex. And each strand helix strand binds one iron sulfur cluster, so the ferredoxin as a whole molecule binds two. So the first thing we tried was to make a symmetric version of this molecule using the tools of computational protein design software that we've developed in our laboratory, consensus-based algorithms that are available. We made a molecule which is essentially the first gene duplication event. So when you had this, for the brown half molecule was duplicated, and you got the entire molecule, adding this little linker right here. So we constructed these molecules, and we found in the laboratory that they bind 4 iron 4 sulfur clusters. We could see this by various types of electronic spectroscopy. And then we measured their electrochemical properties, and the properties, meaning the affinity of electrons, that they had a similar reduction potential to extant to modern ferredoxins. And this was work of Andrew Mutter and colleagues that was published last year. And so now what we're interested in doing is testing whether or not this molecule can also function. So this is that primordial peptide fold where you have a strand, a helix, and another strand. This is the half ferredoxin. And the idea is that 2 of these would come together and dimerize. They would come together and make a pair inside of the cell and look something like this molecule, but now missing this connector over here. And this is the work of Banu, and Banu's going to talk about this in detail tomorrow. So I'm not going to show you much of the data on this, but we are also able to get this molecule to assemble 4 iron 4 sulfur clusters, and its electrochemical properties are also very similar. What's also very exciting about these simpler ferredoxins is that we find that when we put them inside of cells, that they're capable of functioning as electron carriers. So ferredoxin normally functions inside of cells as a shuttle for electrons, moving it from reducing sites like FNR to acceptor sites. And this is work that we've done with our colleagues at Rice, who are part of the NASA collaboration, in particular Ian Campbell and Josh Atkins, and the PI is Joff Silberg. And what they've done is they've set up a pathway inside of E. coli where you're missing the key electron transfer intermediate from a donor and an acceptor. Then we can introduce our design proteins, our models of early proteins, and see whether or not they're able to move these electrons. If they can't move the electrons, then the cell essentially has no energy and it can't grow. But if they are able to move electrons, now the cell is able to grow on a particular nutrient source and reproduce. So we can actually look from the growth curves and see the growth of these. And what's exciting about this is it shows us that these proteins not only have the right chemistry when we look at them in a test tube, but they have the potential to function in a biological context. And so this is the moving into microbial ancestors, the Ma part of ENIGMA, which is covered in theme 2. We're starting to see that these simple molecules can actually function in a more complex environment. And we're very lucky to have this collaboration with the group at Rice to allow us to see these properties. So we can take proteins like hydrogenase, break them into smaller pieces, and then look at these smaller pieces. But these are still, 50 to 100 residues, depending on the molecule that we're looking at. If we really want to think about primordial protein evolution, we should be looking really small, 5, 10 amino acids at the most, because we really want to be seeing things that could emerge spontaneously from let's say, a statistical mixture of polypeptide chains. So from those polymers, what is the chance of making something that's going to bind a metal that's going to have some kind of chemistry? So here's an example of one. This is work that was published 2 years back by John Kim and Doug Pike and colleagues. And here what they did was they took ferredoxin and just looked at the metal binding region. And what they found was that if you look at the structural motif that binds the iron sulfur cluster here and just extend that as a peptide, you can actually make the entire iron sulfur cluster binding sequence out of 12 residues. That peptide was synthesized. We did some electrochemical experiments where we're actually putting these peptides on the surface of an electrode and pushing electrons in and pulling them out to function as electron carriers. And not only that they are actually very robust carriers, that they can be cycled, oxidized, and reduced thousands of times and still have some activity. So these are actually very effective iron sulfur carriers. Very effective electron carriers, despite the fact that we've essentially eliminated most of the protein and we're just looking at the metal binding site. Similarly, in work that Saroj is going to talk to us tomorrow, this is a team effort from Josh Mancini, Saroj Pudel, Doug Pike, and Alexei Tyrishkin, where they have been looking very closely at the nickel binding site of hydrogenase. And they found from a number of designs that only are 8 to 12 residues, so very small, sorts of things that you could imagine emerging spontaneously, that these peptides, when they bind nickel, they're able to produce molecular hydrogen. And not only do they produce molecular hydrogen, but they do it at efficiencies and activities that rival natural proteins. So they're actually not just abysmal catalysts, they actually have very good catalytic activity. And so what we're taking away from this is that these small peptides that are informed by our understanding of how these metal binding proteins have evolved, these small peptides by themselves are actually quite effective catalysts or electron carriers. And this is surprising because there's sort of this paradigm in protein evolution where we imagine that the earliest proteins were probably very inefficient. And over time, over billions of years, what nature has done is taken these inefficient enzymes and made them better, made them more specific, made them faster. And in some cases, that may be true. But what we're finding is that for these core processes in metabolism, the smallest, simplest peptides actually were very effective molecules. And this makes a lot of sense if we think about what evolution would have needed very early on in bringing proteins into a biochemical process. The fact that you have molecules that are very effective means that they would be ripe targets for selection. Some work that will also be discussed tomorrow, so I'm not going to go into too much detail, is in addition to thinking about proteins that we see on Earth and inferring their ancestry from analyzing their structure, we're also using the tools of protein engineering to make novel folds that have no natural counterpart, to see whether or not they can bind metals that are critical for electron transfer and for catalysis, and then whether they function as electron carriers, function as catalysts, and so forth. So we'll hear a lot tomorrow about this particular protein in collaboration with Dror Noy in Israel, and Banu is going to present this. And then we're going to hear about this design as well in collaboration with Michael Hecht at Princeton, and Saroj is going to present this work. All right, so I want to just end on this network again. Remember, this is a functional network. but we're now using this to make evolutionary inference. And we're really just sort of at the start now of mining the data that is in this network and understanding it better. And in collaboration with Lauren Williams at Georgia Tech, we're thinking about the evolution of folds, extracting from this symbolic network and comparing it with his trajectories of protein fold evolution from the ribosome, thinking about the constraints of metabolism and translation, mapping metabolic pathways. This is something that I'm looking forward to the next seminar about, thinking about how we can map the evolution of metabolism onto this electron transfer network. We have a number of summer students who are with us for today in the symposium, who are starting to map electrochemical properties measured or computed onto this network so we can actually see the redox gradients in this network, and then connections with the other themes. So you'll hear from Jana Bromberg some of the work that's going on building real phylogenetic trees of metalloprotein evolution, where the connections are from evolutionary inference. And we're looking now for homology between this functional network and those evolutionary networks to see whether or not, to what extent, we can extract evolutionary information from this. And then finally, in one of the talks tomorrow from theme one, you're going to, from Kenneth McGinnis, you're going to be hearing about how we're trying to map chemistry of minerals onto the chemistries of these different fold types to see whether we can make connections between chemical constraints on the evolution of the geosphere and chemical constraints on the evolution of the biosphere. And there's already some very exciting patterns that are coming from that. So we'll end there. And I don't think there's much time for questions at this point, maybe one or two, and then we can move on to the next seminar.